Okay, now we'll start with the review for microbiology. This is going to be for exam 3. Some things that you need to know for the exam. You should know the definition and what is meant by non-specific resistance, what that relates to. When we talk about non-specific host defenses, there's an aspect referred to as the ciliary escalator, Now, we discussed that in A&P 2. For the benefit of those in A&P 1, the pulmonary system, there is epithelial tissue. A&P 1 knows most of this, but there's epithelial tissue that lines the respiratory tract. The epithelial tissue is pseudostratified ciliated columnar epithelium. Now, all of you, even A&P 1 and A&P 2, know that. There's cilia there. Cilia has a mucus layer that lies over the top of it, and so when bacteria and viruses from the external environment come into the respiratory tract, it gets caught into that mucus layer, and then in a wave-like fashion, the cilia throws that mucus out of the system. When it throws that mucus out of the system, it's actually a form of a mechanism by which organisms are removed. So in your textbook, they describe the ciliary escalator, and so you should be familiar with what the ciliary escalator relates to, and I just described it for you there. Lysosomes, you should know what secretions you see lysosomes in and why lysosomes are important in removing microorganisms. You need to be familiar with the complement system. Remember, I told you the complement system aids the immune system. There are different types of complements. I didn't talk about them much in the lecture, but you do need to read about them. Read about complement C3A. Complement C3A. Recognize what it does. What types of symptoms do you see when the complement system is activated, such as increased blood vessel permeability, acute inflammatory response, and you should know the acute inflammatory response, dilatation of blood vessels, histamines, mast cells, inflammatory cells coming to the surface or coming to the area of attack when an antigen enters into the system. So you should know what the activation of the complement system is, specifically activation related to the complement C3A. You should know, remember we talked about catalase positive type of enzymes that particular bacteria can produce, and superoxide dismutase is another type of enzyme similar to what we see in catalase. Catalase produced by bacteria has a specific role. You should know what the role of that enzyme is for that bacteria. Let's see. You should be familiar with, as I said earlier, the pathway, the classical pathway for complements. Complements aid the immune system. Complements typically are initiated by an antigen-antibody reaction. And so you should be familiar with the classical pathway for complement activation. Complement activation typically is initiated by antigen-antibody reaction, but you need to read about that to be able to answer the question related to that on the exam. You need to know the characteristics of inflammation. The characteristics of inflammation. We already talked about redness, local heat, pain. You should know those general characteristics. There are more of them. You need to read about that. I didn't talk about the inherited deficiencies when it relates to the complement system, but you should know what happens when a person has a deficiency in the C3 complement. I already told you to read about C3A. You should know what happens if a person has a deficiency in the C3 complement. You should know the term complement fixation. And we talked about some of the, that illustration I showed you in the slide showed you all of the components of complement fixation. If you remember, we talked about opsonization. We talked about cell lysis. It's on that illustration in one of the first lectures that we did today. So you should be familiar with what happens in complement fixation. Again, you need to know what happens in inflammation. Wow, 3 questions about inflammation. 3 questions about complement too. Normal flora or normal microbiota? What do you need to know about that? Characteristics of normal flora, normal microbiota. If you remember before last week, we talked about how normal flora competes with invading microorganisms for the same energy source, and so they help the body in that regard. Excuse me. So you should know the role of normal flora. I already told you in parasitic infections, we see a response by IgE. We see a response by eosinophils. These are all cells that come into the inflammatory network. You should be familiar with that mechanism in parasitic infections. So you need to look up IgE. You need to look up eosinophils, specifically as they relate to infections related to parasitic organisms. You need to know the term margination. Margination. And I already told you to look up inflammation. There's another question about inflammation. Wow. You should know the following types of immunity. You should know what innate, I-N-N-A-T-E, innate resistance. You need to know what naturally acquired active immunity, naturally acquired passive immunity, artificially acquired active immunity, and artificially acquired passive immunity. Yeah, innate resistance, naturally acquired active immunity, natural acquired passive immunity, artificially acquired active immunity, and artificially acquired passive immunity. That was all in that first lecture that we did today. And I'll go a step further. One immunity you get from vaccination, one you get by getting transferred an antibody, say, from your mom. Another type of immunity you can get by means of an injection. So those are the basic types of immunities that we just discussed. You need to know about plasma cells and what activate plasma cells to work. You need to know the basic definition of an antibody, and I gave you that in the presentation today. You need to know what happens to the antigen once it's coated by the antibodies. Makes the antigen susceptible to a specific type of an attack, so you need to be familiar with that. You need to know what happens when an antigen becomes coated by the antibodies. It makes the antigen susceptible to a specific type of an attack. You need to be familiar with that. And you need to review that slide presentation again on IgG, IgM, IgA, IgD, and IgE. And I gave you just a little bit of snippets about, is that the right word? Snippets? 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 I don't use that word that often. What do you call it? Snippets. No, it's snippets. It starts with an S. I know it starts with an S. Snippets. You've heard it before. Snippets. I gave you little synopsis of each one of those antibodies and why they were important. Snippets. It's snippets. I'm on, yeah. Snippets. Snippets. Tidbits. Snippets. Okay. About IgG, IgM, IgA, IgD, IgE. IgE, you see that in parasitic infections, remember? IgG, very common, remember? Those types of little snippets that I gave you. Tidbits, snippets. I like snippets about each one of those. Which one of those do you find in secretions, saliva, tears, that sort of thing? We talked about fungal infections just a little bit. Just remember, fungal infections, you got to treat those with antifungals or fungicides. Bacteria you treat with antibacterials, right? Viruses you treat with antivirals, okay? Just general category of medicine. So if I ask you, how do you treat a viral infection, hepatitis A, you don't treat that with an antibacterial because it's a virus. If I ask you how to treat sporotrichosis, which is a fungal infection, you treat that with an antifungal. You don't treat it with an antibacterial. We talked about spore trichosis last week. If I ask you to treat a bacterial infection, you treat that with an antibacterial. So that's the point that I'm trying to make. We talked about smallpox last week. You should be familiar with smallpox and the treatment for smallpox. We talked about thrush and vaginitis last week, what the most common organisms are that causes thrush and causes vaginitis. We talked about herpes infection last week. Herpes gladiatorium we talked about. You need to know how herpes is transmitted. We talked about varicella zoster last week as well. Talked about how varicella zoster, what it presents with, what the signs and symptoms are. You should be able to differentiate between varicella zoster and smallpox. Different presentation in both of those. You should know the mode of transmission for chickenpox, smallpox, and German measles. How it's transmitted. Through blood, through respiratory droplet, through direct contact. How is it transmitted? German measles. Yeah, German. Instead of the French measles, the German measles. I'm sorry, I want to pick on the French right now. They're having some problems. Yeah, and should know the presentations of smallpox versus chickenpox. We also talked about scabies last week. Need to know about scabies. What type of organism is scabies? How it's treated. You need to know the general characteristics of Staphylococcus food poisoning. You need to know some general characteristics about Helicobacter pylori. We talked about those today. You need to know the most effective treatment for treating most gastrointestinal infections. Let's see. Need to know about salmonella. Need to know what animal do we see containing salmonella most often, how you get it, how you treat it, what the morbidity and mortality rate is. Different forms of hepatitis, hepatitis A, B, C, D, and E. You need to know what's the most common way that you would get hepatitis A, most common way that you get hepatitis B, most common way you'd get hepatitis C. And to get hepatitis D, there also has to be a comorbid infection with one of the other viruses. You need to know that. Remember, one's caused by fecal oral, the other one's caused by blood transfusion. I already talked about salmonella. Need to know the organism that you see from contaminated pork. We just talked about that. Let's see, talked about varicella. You should know those Vibrio diseases that you had to do on your lab. There's a question here asking you to identify which organisms can cause gastroenteritis. So you need to be familiar with those organisms from the lab. We already talked about the location of microorganisms through the gastrointestinal tract, beginning at the mouth, all the way through the large intestines, where most of the organisms exist in the gastrointestinal tract. And then we also talked about the effects of mumps in the gastrointestinal tract as well. Okay? That's the lecture portion. Yes, question on the lecture portion of the exam. There are 50 questions. Yes, 40 minutes to do that. Because some of them are questions you answer real fast. It won't take you a full. Now for the lab. You had your first lab. I'm sorry, I don't necessarily have the order here of your labs, but you should know what gastroenteritis is, what organisms can cause gastroenteritis. You should be able to define the emetic syndrome. You should be able to define the diarrheal syndrome. You should be able to tell me ways that you can prevent food poisoning by proper food handling. One of the organisms that you discussed on that labs had a toxin involved in it. You would need to know what the symptoms of being exposed to that type of toxin is. There is also a lab that talked about acanthamoeba, organisms. Where do you find this organism? Where does it exist? You should know some of the diseases related to acanthamoeba. It talked about 2 specific types of diseases that were related to acanthamoeba, and you should be familiar with those. You should know how you can culture an amoebic organism. It talked about using a specific type of an agar. Talked about Naegleria species in the lab as well, where Naegleria species was located at, and where in humans does it cause a specific type of an infection, and what type of an environment would the human need to be in to contract this infection. You should know this term when it relates to viral infections, called cytopathic effect. I think I've tested you on that before, but you should be able to list the cytopathic effects of viruses. And then the labs also discussed about the dust mite, dermatophagoids. Where do you see them exist at? What is their impact on humans? What types of diseases do we see caused by these mites or these dermatophagoids? How do you treat mites or dust mites? If there's a cure for it, what is it? And then I believe your last lab talked about the head louse or head lice, Pediculus humanus capitis. You need to know how these are transmitted from person to person. What steps need to be taken to rid a person of the disease? The relationship between body lice or head lice and body temperature, and why that's important. And then you need to be able to list the organisms that are related to the gastrointestinal enterics. There were a list of diseases in that PowerPoint presentation on the gastrointestinal enterics. And just as in the lecture, we talked about salmonella, what food industry is commonly infected with salmonella. Now in Shigella, you need to be familiar with a concept called pseudomembrane disease, pseudomembranous. What happens, and I'll go ahead and explain this. In Shigella, what happens, the organism forms a coating around the wall, around the bowel wall, inside the large intestine. So the large intestine is primarily responsible for absorbing water. If you have a pseudomembrane around the intestinal wall, the large intestine can't absorb water. If you can't absorb water, you have diarrhea. Does that make sense? So when I ask you to explain to me the concept of pseudomembrane or how that's related to Shigella, you should be able to describe how the bacteria forms a coating. And it's explained in that. But you should explain it the way it's explained in that PowerPoint. Basically, covers the intestinal tract. Maybe it's not explained in there. Explain it the way I just explained it now, in case it's not in there. But basically, the organism secretes a coating around the intestinal wall, and the large intestine can no longer absorb water. And then you should know what organism leads to rice water stools. And then how would you treat the organism that causes rice water stools? That's it. May the force be with you. I don't know in the lab yet. These are the questions I'm gonna take your questions out of the questions I just reviewed for you, but I don't know how many questions. So I think I gave you 20 minutes on the lab. Who asked you? Didn't you ask me a question? I think it's 20 on the lab, so probably be 15 questions, because you have to write some on that one. Here you have to type it in, so I'll give you a little more time per question. Okay? Very good.